Good evening, ladies and gentlemen, and welcome to a FreeThought Fort Wayne educational presentation. I'm Robert Brandt. I'm this year's president of our group. And could I ask you now, as happens everywhere when you're in a public forum, please silence your cell phones. FreeThought Fort Wayne is a cultural and educational, nonpartisan group dedicated to leading ethical lives that aspire to the greater good of humanity. We are also affiliated with the American Humanist Association. I'm sure many of you saw the table at the back of the room. We have some free literature back there to examine, as well as a sign-up sheet if you wish to allow us to contact you about other future events. There's also a membership form if you would like to join us. Your greeter, Karen Meisner, can assist you after the presentation. Also, there are some complimentary snacks and water if you care to socialize with us after the presentation. There will be an opportunity to ask questions after the presentation. The only request I would have, since I tend to be kind of wordy, and a lot of people like to make speeches rather than ask questions like me, please, if you do have something to say, please end it with a question mark. It is my privilege now to introduce our speaker, our very own Christopher J. Rex. As you can tell, Christopher is an adjunct professor of biology at Ivy Tech Community College in Fort Wayne. And his subject is DNA and Me, How Genetic Testing Improves Evolution. Please extend him a warm welcome.
I guess I'd like to start with why in the world did I choose a picture of the sky for the beginning of this talk. And the thing that comes to mind is the one thing that really gets under my skin when it comes to public education. And being in the public education sector, not only at the college level, as I've illustrated here, but also working at Fort Wayne Community Schools, one of the things that bothers me is seeing this kind of perspective being taken on evolution, which is to say, a more creationist point of view. The issue with taking that as an alternative to evolution and thinking that some sky god created all life is the fact that it's not terribly informative. It's not terribly predictive. And it doesn't honestly help us in any way, shape, or form when it comes to certain things that we do in the scientific realm. And this presentation, I hope, will help illustrate that for you. And so that's why I've chosen a picture of the sky, because I like to think of heaven and angels and everything when I see a pretty picture of the sky. So that's kind of the tone that I'm coming from as far as why we're looking at DNA, why we're looking at genetic testing, and how genetic testing even relates to evolution in the first place. So just to give you an idea of how this lecture is going to go, first we have to talk about DNA, because that's kind of the whole topic of the talk here. And then we can actually relate it to genetic testing, as far as what we're doing when we take a sample from you and analyze your DNA. Then we have to relate that back to the theory of evolution, kind of fleshing that out a little bit, and even break it down into the micro and macro evolution that some people like to talk about along the way. Then, of course, we have to talk about evolution of various things. Now, you see some very odd things on here, as well as some very usual things on here. Like evolution of shapes and colors. Well, this is sounding more like an elementary class than it is anything else. But I promise it does relate to the evolution of primates and humans as well. Then, of course, we have to talk about the controversy a little bit, and then we'll finish up with some questions if you have any by the end. So, first off, we have to talk about the boring stuff. All right. For those of you who don't remember your freshman biology class, which I'm sure is pretty prevalent among the audience at the time, because honestly, I don't remember much from my freshman biology class. I only remember this stuff because I get to do it every day. So when we talk about DNA, we're talking about deoxyribonucleic acid, which is a big fancy word for this. You guys are familiar with this image? You know that this represents DNA. Now we're going to compare this to the other type of nucleic acid or genetic material, which is ribonucleic acid. And that's kind of represented here. Most people aren't accustomed to seeing that. Two reasons for that. One is the fact that this is typically single-stranded, whereas DNA is double-stranded. Secondly, RNA has certain different uses than DNA does. So we're going to talk about those here in a second. But when I really talk about deoxyribose versus ribonucleic acid, the only chemical difference between the two is that deoxyribose doesn't have an oxygen. And I'll show you a picture of that here in a second. When looking at the structure of any nucleic acid, it's really composed of three different parts. First off, you have the 5-carbon sugar that I just mentioned, whether it be deoxyribose or ribose. Right here, we're talking about deoxyribose. So at this particular location, it does not have an oxygen. Compare that to ribose, which does. Not like you care, but this is the difference. The other difference, or the other thing that's in common actually between DNA and RNA is the fact that they both have phosphate groups and they both have nitrogenous bases attached to them. So we see a phosphate group here and a random nitrogenous base here. Now nitrogenous bases are another point of difference between DNA and RNA. The fact that DNA has thymine while RNA has uracil. Other than that, they both have adenine, which is A, guanine, which is G, and cytosine, which is C. Once again, not terribly interesting. What is interesting is how they bond together. Okay. When I showed this previous picture, you basically kind of imagined a ladder with these rungs that was twisted up. Well, these rungs are actually not just one thing. They're actually two things coming together. And those two things are the nitrogenous bases. That's because the nitrogenous bases bond together with their counterparts, so to speak. So A always bonds with T, C always bonds with G. Now, obviously the exception is in RNA, A bonds with U, not T. Getting into how this actually relates to us, we are going to be talking about DNA. The reasoning for that is because RNA is only present in a few organisms, most notably the more primitive organisms, things like bacteria, and even things we don't typically consider organisms, viruses. We're not going to be talking about that. We're going to be talking about us, because everything in us originates from DNA. Just to show you how that works, we have a strand of DNA, all these A, T, C's, and G's all mixed together here. And what we do is we actually take DNA and transform it into a different form. And you may be like, well, why do we even bother with this whole transcription step of converting DNA to RNA? That seems to be backwards. Well, that all comes down to the fact that DNA is locked inside the nucleus of your cells. DNA cannot leave the cell. And so somehow we have to get that message out of the cell. And that somehow is through RNA. Once it leaves the nucleus, I'm sorry, not the cell. Once it leaves the nucleus, then we're able to translate, which is to say, take the information in the RNA and convert it to a new form that's actually useful. Okay? The RNA itself is not terribly useful. It doesn't do things within the cell per se. The proteins, however, proteins are what represent basically every single part of you that is useful. And it's composed of multiple different types of amino acids. All these different things really come down to how DNA is broken down as far as the information is concerned. Bits of DNA that correspond to the creation of a single protein are called genes. Proteins, once again, are just polymers of amino acids, but they're all the functional parts of you. So if we just think about the whole process here, you have your cell with the nucleus filled with DNA. These are in chromosome form right now. If we think about these pieces of DNA being copied into RNA, in this particular case mRNA, that will then go outside the cell, be translated into amino acids, which form a protein. Like, oh my gosh, this is so much stuff. Well, there's one more thing we have to talk about, and that is how the nucleotides themselves are not one-to-one correlated with amino acids. If you have 3 nucleotides, you will only form 1 amino acid. And that's because of this whole codon thing. Which is to say, when you read 3 different letters, any 3-letter combinations on this chart of 64 different possible combinations, when you read any 3 letters, you will create an amino acid of some kind. But you'll notice some of these amino acids, like this threonine right here, he has 4 different 3-letter combinations that code for him. So when we look at this, this is an incredibly redundant system. Which is to say, you can almost think of this as being different email addresses that you have. All 4 of these email addresses all lead to you. But there's still 4 different ways to contact you. Okay, so we can almost think of that as being like the genetic code. And this doesn't just apply in humans. This actually applies universally, which is to say, this same code codes for the same amino acids, whether it's inside you or inside a bacterium. It doesn't matter. It'll code for the same exact amino acid. Now, if we think about how these nucleotides change, we normally think of the word mutation. Right? We think of it as a negative kind of event. When really all mutation refers to is a change. That's all it means. And given the nature of these codons, that change is not always bad. Okay. If we just change this last letter in this codon from U to C, it doesn't really matter. We still create threonine. There is no change in the resulting protein. So when we think about the different outcomes from mutation, you can either have no change, or you can break the protein. Create something that's not functional by converting it to some sort of nucleotide which generates some different amino acid which completely breaks the whole function of the protein. Or we can create something different. That something different might just be different, or might be beneficial, or both. So we have to keep in mind that mutations aren't always a bad thing. And the different types of mutations kind of determine how bad or good the outcome is. You know, sometimes we delete, insert, substitute, or do something really crazy. Which is to say, if I physically removed the C right here, I would shift the reading frame entirely. Of every downstream amino acid, I would change them all. And that's what we call a frame shift. That's really bad. Typically, that always results in a non-functional protein. Backing up and looking at the larger view, we were talking about chromosomes. And we were talking about genes on those chromosomes. Well, different forms of a gene are called alleles. Which is to say, if I looked at a pair of chromosomes here, they code for the same type of information. In this particular case, we're talking about an allele for blonde hair. Now, this particular chromosome I got from my mom. Let's just say, because I clearly don't have blonde hair, not like you could tell anyway, but I don't have blonde hair. Then we have another allele from my dad. If it's also blonde hair, then I probably have blonde hair. However, if we think about this case here, we have an allele for blue eyes and an allele for brown eyes. Now, funny enough, this blue eyes allele arose from a mutation about 8,000 years ago. So that's kind of cool. Very interesting that we can trace it back that far. But that's how these alleles happen. That's how they generate change in a population. It's through this mutation process. If we think about what a chromosome is, your DNA actually normally looks like this. A thready mess of a plate of spaghetti that Chris makes. Because I'm not a cook. However, when we think about chromosomes, they're these very nice, beautiful structures that sometimes look more like a tightly folded X together. This arises because we actually wrap this thready stuff using histone proteins. And so those histone proteins are very, very important when it comes to the regulation of those genes that they're wrapping up. We're not going to get into that, but we'll come back to it here later on. Now, looking at the fun stuff that you actually can see for yourself with your own eyes, we can actually start looking at chromosomes themselves under a microscope. Now, granted that we do special staining processes and such, that's when you can start seeing the fun. But if we think about the meaning of chromosomes in general, the number of chromosomes really doesn't matter. Okay, humans have 46 chromosomes, exist in 23 different pairs. Right, so you get one set of chromosomes from your mom, one set of chromosomes from your dad, and there you go, you have 46 chromosomes. When we think about what 46 means, it really doesn't mean anything. Those are just different structures that have been folded up from your DNA. Doesn't necessarily mean that we're better than any other creature, especially when we compare it to this adder's tongue fern, which has 1,260 chromosomes, which is ridiculous. Plants have a funny notion of duplicating their chromosomes for various reasons we're not going to go into. But needless to say, this does not necessarily make this plant any more complex than us. So the number of chromosomes really doesn't have meaning. What matters is the information contained in them. And that's really what we're going to try to focus on here. Now, if we think about humans, the thing that determines what gender you are, for the most part, is really dependent on these 6 chromosomes. If you have X and X, you're female, X and Y, you're male, generally speaking. As far as the rest of you is really determined from the other 22 pairs of chromosomes, which we call autosomes. And this is a really nice picture showing all 22 of those. Looks cool, whatever, but there is a whole lot of information contained here. And we're going to start exploring some of that information because this is the information that's really going to tell you where you came from. And that's where genetic testing comes into play. If we think about genetic testing, it's come a long ways. Just in the past 15 years, it's gone from costing thousands of dollars down to $99. Or $199 if you need a health report, which is always a good thing. But if we think about how they do these types of genetic testing, they really don't sequence your DNA, like some people think. Okay? We had the Human Genome Project a number of years ago, almost 15 years ago actually, and they actually went through and sequenced a bunch of DNA, looked at all the genes, and discovered all this fun stuff. That's not what they do. They don't care about that. What they're doing is looking at your genes, which is to say those long stretches of nucleotides that code for a single protein. That's what they're doing. And that's how they can do this 10,000 times faster than normal genotyping techniques. And they do this by looking at hundreds of thousands of locations on your DNA, which is kind of nice. Especially when you have 3.2 billion nucleotides. Hundreds of thousands seems like pennies compared to that number. And that's why they can do it so much faster. That's really nice. The one that I've done in particular is 23andMe, hence the name of this presentation, DNA and Me. And so what we're going to do is we're going to kind of look at how they do things, because how they do things is actually how a number of different genetic testing places do in this modern day and age. They don't need to draw your blood at all, which is great for me because I hate needles. I really do. It's like nails on a chalkboard magnified by 100 when it pierces my flesh. I can think of nothing but that. I hate it because it's my only weakness in the world, but thank goodness we have saliva and skin samples. Of course, I say only weakness in the world next to my wife. But anyway, it's great. Now all you have to do is order this kit, have it delivered to your door, and spit it in a cup. If you chew tobacco, it's natural for you. It's great. So all you have to do is just spit in there, send it off. A couple weeks later, you get your results. And it's awesome. So convenient. Now, of course, nowadays, what we namely use genetic testing for is determining, of course, your ancestry, which most people end up being curious about one point or another. But if you're a fan of television, which I'm kind of a television junkie myself, and I grew up on the allure of Jerry Springer and The Maury Show, which explored a number of different instances where people didn't necessarily know who the father was. And this was a great way to do it. Very, very simple genetic technique where we can look at and analyze who the daddy is. We can take the mother's DNA sample, the child's DNA sample, and the father's, supposed father's, DNA sample, and compare them. But when we do this, we're not looking at all the genes. Okay? We're not looking at those hundreds of thousands of locations. We're actually limiting it in this case. We're doing what's called DNA profiling, which is a very quick and easy method of comparing DNA. Reason why is because we're only trying to see if they're different. And it turns out you only have to look at 13 different locations in human DNA to figure out if two people are different or not. So we're not trying to figure out what your genes are. We don't care what hair color you have. What we care is how many repeats you have. There are these short tandem repeats, or stars. So it's a segment of DNA, let's just say it's AATT. It's a segment of DNA that repeats a lot in your DNA. Doesn't necessarily code for much of anything, but it's a great way to identify who you are and distinguish you from other people. And so what we can do is we can do this procedure called a gel electrophoresis, where it lines everything up based on how many repeats. And so if you see, well, the mother has some genetic characteristics in common with the child, which is good because the mother should know that the child is hers. But then we can line up the father and see, well, do any bands line up with him? And we see this band does, that's good. This band does, that's good. And this band does. So guess what? You're the daddy. That's how easy it is when it comes to very simple things. For our purposes, though, we want more information. In order to do that, we have to keep in mind what we're comparing. What we're comparing is the DNA inside your nucleus, and we are analyzing the types of genes that are found within them. Because the types of genes determine you and all of your proteins and exactly what you look like. Also what we can do, though, is there's this other location inside your cells. We talked about the nucleus. That's typically referred to as the control center of the cell. There's another location in your cell called the mitochondria. Amazingly enough, this tiny location is the whole reason why you're breathing oxygen right now. Without mitochondria, you would not be breathing oxygen. It's the mitochondria that allow you to take in oxygen, break down glucose, and exhale carbon dioxide. This tiny little part in your cell. Crazy thing is that this tiny little part also has DNA of its own. Only 16.5 thousand nucleotides in length, which is chump change compared to the 3.2 billion that we were talking before. Other neat thing about this is that it's only passed from mother to child, which is to say, sorry guys, the little sperms that you contribute have 0 mitochondria. They've been stripped away. The cool part about this is that it has a very, very slow mutation rate, so we can track it back in your ancestry very, very accurately to a very long time. It doesn't mix and get all swished around like this nuclear DNA does. And we'll talk a little bit more about mitochondrial DNA here in a second. A couple things I would like to mention. One is the genotype. That's what we talk about as far as your genes. Phenotype is what happens from the proteins that those genes make. If we think about your genotype, even though you might have a gene that makes you predisposed towards becoming addicted to cigarettes, it doesn't necessarily make you addicted to cigarettes. The reasoning for that is that we have a lot of environmental influence on your genes and how they're regulated. So even though I might have the potential to be a great piano player, not having a piano, I will never realize that potential. And you know what? I've come to terms with that. I'm moving on in life. But it also applies to identical twins. Okay? They have the exact same genetic makeup, but they can be very, very different as they age. And that's all because of this environmental influence. So if you're thinking about this nature versus nurture kind of paradigm, it's really both, all the time, constantly throughout your life. So if you have a health report that says that you have a 20% higher than average chance of getting pancreatic cancer, that doesn't mean that you'll get it. It just means that people with those genes get it 20% more often than the rest of the population. Is this something to watch out for? Yes. Does it mean that you'll get it? Not necessarily. So always keep in mind that these are not absolute types of things that are damning. Also keep in mind that a number of these genes play with other genes to make up you. Okay, when it comes to your eye color, when it comes to your skin color, we have numerous different genes that all provide inputs, which is why when you look at somebody's eyes, even eyes that look as bland as mine, you will see flecks of other colors in there. Eyes look brown. When you start looking at them, they have flecks of blue and red and green, all sorts of colors. Funny enough, if you look close at my goatee, it also has flecks of other different kinds of colors, which is ridiculous. But luckily, it's turning more gray every day, so that'll be fun. If we think about how this relates to your ancestry, though, that's where it becomes fun. Because now we're trying to figure out where you came from, where your ancestors came from, and how inbred you are. Which is to say, I'm very inbred. So if we think about my genes, I am 99% white bread as can be. All right, I'm European, except for a dusting of East Asian. Makes me feel special. It's okay. But if we think about how this whole thing operates, we can do this because we can go back as far as 1 million years into the past and analyze DNA. So all those fossils of different humanoids that have existed, we can track their genetic makeup and see how related they are to you and where you come from. We can do that. Unfortunately, when it comes to more recent splits, it may not necessarily be as informative. The reasoning for that is because maybe when you heard that your great-grandma came from Ireland, as was in my case, she may not necessarily have been 100% Irish. So keep that in mind. But I'll show you a graphic here of what I'm kind of talking about. If we think about where I came from, I'm pretty boring for the most part, but 2/3 of me is British, Irish, French, and German, which is to say, Northwest European. The rest of me is basically everywhere else in Europe. Imagine that. So if we think about a map kind of showing where I'm from, the colors mean nothing, by the way. It's really just kind of localizing locations. It's not intensity related at all. But like I said, namely here, but really all of this, which is to say very limited. But the cool part is that we can actually look at and guesstimate how many generations back your ancestors were from certain areas in this map. For instance, my most recent ancestor in the past 100 years or so was British and Irish, 100%. 150 years ago, I had an ancestor that was 100% French and German. About 6 generations ago, that's when it just becomes slop. Okay. A little bit of here, there, and everywhere. The only interesting breakdowns are with the Balkan and the Italian, which is to say my mother's side has the Balkan, and my dad's side has the Italian. Otherwise, both sides of my family have everything here. So once again, with the exception of 2.5% of my genome, I am highly inbred from Europe. I love this picture so much because this actually relates to the other picture of chromosomes that you just saw. Only now, every chromosome is laid down on its side, and you get to see the colors and how each of the colors relates to each of the different locations over here. Now, unfortunately, I don't know why 23andMe decided to use shades of blue for everything, so this kind of looks like a disaster. But what's nice is that online, as you hover over each of these parts, it'll tell you what parts of your chromosomes came from which locations. That's really cool. You know, for instance, chromosome 17 here, this contains both French and German from both my parents. This is entirely 100% French and German, and it's from both parents. Which, once again, inbreeding. Awesome. The fascinating part, though, is this little guy, that East Asian part. Heck, what is going on with that? That is that tiny little orange fleck way up there. Like, huh? Aliens? I don't know. That's really strange. Unfortunately, you only see the X chromosome down here, though. You don't see the Y. And that's because we can use the Y for certain other important bits of information, such as where all of my daddies lived and came from. Which, once again, Europe. The cool part, though, is just thinking about how the majority, about the 50% range, most of them originated from the area that would later become home to the Vikings. So I can pretend like I'm descended from Vikings. But unfortunately, we're talking about 28,000 years ago, which is about 27,000 years before the Vikings. Unfortunately.
I'm sorry, Chris. Wasn't the universe created 6,000 years ago?
Not according to science.
No.
That's the unfortunate thing.
About that? Does this, in fact, show us something about that?
It basically shows us that that science can really determine the age of not only the Earth, but also the age of our ancestry. Cool. Yep. And so when it comes to the maternal line, we can be a little more accurate with it, once again, because it's mitochondrial DNA, because of the fact it's not mixing with anything. And what we really see is that my mom's side of the family kind of originated over here, kind of in the northern Middle East. Okay. And they kind of spread from there, which is interesting because her line goes right through the Balkans, which is why I get the Balkans from her. If you look at my dad's line, his line goes down into Italy a little bit, which explains why I get my Italian side from him. But if you look at this a little more carefully, we can start doing some fun things with this. We can start seeing how all human mothers originated, this is really hard to see on this, but this is the continent of Africa here, and all human mothers originated from this point, L. That is to say that this is the cradle of civilization. This is the veritable Garden of Eden. Unfortunately, many creationists think it's way up here. They're only off by a few miles. Distance between South Africa, at least. The reason why this is because we can trace mitochondrial DNA in all the fossils, in all the humans that existed in all these areas, and see that they all descended from this single point. About 150,000 years ago. And we call this original mother mitochondrial Eve, for the simple reason that it's fun to poke fun at religion. No. The real reason is the fact that this is truly where all humans came from. If we track the Y chromosome, we can track it back to the Y chromosomal atom, so to speak, and we can do that back to about the same time. These numbers were gotten by independent researchers using different methods, and they just about agree entirely, which is really quite incredible. So the birthplace of humans was Southeast Africa, and the time was about 150,000 years ago. So much longer than the 6,000 years, unfortunately. Now if we think about evolution in general, and what this means, what this must mean if humans originated that long ago, we have to think about what even evolution entails. So the theory of evolution is talking about how every living thing on this planet is just the result of small changes over time, and how they all originated from a single common ancestor. That's really what it's talking about. Every plant, every animal, every fungus, every bacterium came from one creature. But as far as how that one creature came about, that's where we have abiogenesis. That's different and distinct from the theory of evolution. If we think about what abiogenesis stands for, abio basically means not life, which is to say the origin of life from non-life. And when we look at it biochemically and think about how to predict it, it's really inevitable, given the right conditions and chemicals that are present. And most people have taken this to be like, oh yeah, well, if you find another Earth. Well, we found a number of different Earth-like planets. And we keep finding more, seemingly every year. But it doesn't necessarily take Earth. We have found complex organic molecules in a number of different places in the universe. Not only different planets, but also asteroids and gas clouds in the middle of space. Things like amino acids and nucleotides. And what we found is that it only takes 5 nucleotides. 5 nucleotides to come together and form the simplest thing that we can call life, which happens to be a molecule of RNA. That is the smallest thing that can exhibit all the symptoms of life. And yet, when we think about what that is and how it works, gosh, that really seems like a lot of stuff to swallow. And so we have to further break down the theory of evolution to further understand how that even works, because this just completely goes against everything that we've heard, right? We somehow heard that you can't possibly form an eyeball from random parts and chemicals. You know what, you're right. But we can certainly form life, which begets more life, which adapts and evolves. And that's where the theory of evolution comes into play, from these 3 different aspects of evolution. We've already kind of talked about the theory of common descent. Everything has to arise from one single common ancestor. But along the way, the genes have to modify. The DNA, or the RNA, as it would be in the first organisms, has to change over time. Now, once again, these mutations can generate different outcomes based on what's going on. But then we have this other weird thing, that environmental thing that I mentioned, that non-genetic changes can also happen. And that's going to change how the genes are even expressed or regulated along the way. So that's really going to impact that too. But we also have to keep in mind that this process of evolution is very, very unpredictable, completely random, and it's not something that produces perfection. I don't know how anyone has ever come to this claim, but some people seem to think that humans are perfect. And even though there are times, as my wife can tell you, that I may think I'm perfect, there are certainly a number of instances where I am not. One perfect instance is the fact that I'm still sick and recovering from a cold. But if you just think about it, no organism on this planet is perfect. So thinking about this idea that evolution produces perfection is just lunacy. It doesn't happen. And thinking that we have an idea of what evolution is going to generate in the future is also just crazy, because it's so random. There is no way to predict it. And there is no way to stop it. You cannot stop evolution. We can spend billions of dollars generating and producing this huge particle accelerator. All right, if any of you are familiar with CERN and the giant particle collider over there, that thing can look at particles and sustain them in these magnetic fields for a lot longer than they actually exist in reality. It can stop a particle that is subatomic and subatomic beyond that. But we cannot stop evolution. No matter how much time, no matter how much resources you have, you will never stop anything from evolving. It will always happen. If we think about how we evolved through a long process. Okay? We are not to say the pinnacle of evolution by any means. But for all intents and purposes, we're the pinnacle of evolution. If we think about the other kinds of creatures and how we group all these other kinds of creatures. We group them in a way that lumps similar things together. First and foremost, we're a member of the eukaryotes at the domain level. We're animals, chordates, we basically have spinal cords and things to protect them. We're mammals, of course, we all know that. But we're also primates. And then we start getting into the more human-like creatures, all the way down to the Homo sapiens that we know and love. But I'm going to be mentioning species here a little bit, so I'm going to go ahead and define that for you as a group of organisms that interbreed and produce fertile offspring. I emphasize fertile offspring because certain instances where we can generate offspring, but the offspring are sterile in the animal kingdom. We're not talking about that. Species is something that can produce fertile offspring. Now, strangely enough, we are a subspecies. So we're actually Homo sapiens sapiens, which is to say, we did have other members in our species, which were wiped out. But some people like to split evolution into two different main categories. Microevolution, which they typically consider to be on a small scale, something that they can witness in the lab, doesn't take very long, might take decades or something, but it's really just representing genetic changes in a population that are significant enough to produce something that we consider different enough to be a different species. So if you think about having a block of wood that's circular in shape and sanding off the sides of it to kind of form this octagon, we can certainly go to a carpenter or somebody, a woodworker, and watch them do that right in front of our eyes. We can see that. That's not hard. However, macroevolution operates on a time scale that's many times that, many times longer than that. We're talking about many millennia. All right? These are larger scale trends that apply not necessarily to the species level, but to every level above it. We're talking about different genera, families, and orders. Okay? Really different organisms. And so we're kind of talking about taking a circle and making it into a star. Now I know there are probably bakers in the audience that are going to sit here and say, well, I can take a circle of cookie dough and make a star just by taking a cookie cutter to it. You'd be right, but we're not talking about cookie dough. We're talking about wood, right? Stay with me. A block of wood is very, very difficult to take to a star in one step. We have to do many steps to do that. So when we have this example presented, many people are going to be like, well, that's impossible to do in one step. But we can do it if we take multiple small steps. So if we do kind of what we did before and sand off the edges to that circle and make an octagon, then we can sand off some more edges, make it into a square, take that square, make it into a hexagon, sand off some corners, make it into a pentagon. Then we can make some inside cuts, straight line cuts, and we can generate a star. This is how we can go from a species in a certain genus to a whole new genus, by going through all these intermediate steps. Which is to say, this takes a long time. A long time for any carpenter or woodworker to do. Another way that we can kind of think about this whole concept is relating it to a color spectrum. And thinking about how the color blue or purple kind of evolves into red over this particular color spectrum. If we think about that and relate it back to our micro and macroevolution, we can think about macroevolution as being difference between yellow and red. You're like, yeah, okay, I can call that yellow and red. But how do we get there? Well, how we get there is through all these different shades. Shade into orange, different shades of orange into different shades of red. It's very, very hard to make it through that process in a short time frame. What's even harder is picking any point on this color spectrum and being like, oh yeah, this is aqua. it's kind of a mix between aqua and light green. It's very, very hard to define that color, especially if we say, well, what's the difference between these two spots? One's 50% green and the other's 52% green? I don't know. But that's kind of what we can see, though. We can see shades of color change over time when we're looking at animals in our world right now. We can see the color shades change. But it takes a lot of change to be able to call them different colors. We would still call both of these points greenish blue. Further, when we think about something that often plagues scientists, which is to say the gaps that exist in the fossil record, we can represent those gaps as black bars on this color spectrum. And the question remains, even with these black bars in place, can you still tell that blue is transforming into green? And the answer is yes. The presence of the black bars doesn't negate the overall picture. It doesn't matter. We have enough information to make the extrapolations that are perfectly logical. And so the gaps in the fossil record are just gradually being filled in. And they get filled in at exactly where these points are. That's okay, because it all fits together. If we relate that to our evolution, we can think about us coming from a common primate ancestor, spawning into all these different kinds of monkeys and great apes, and eventually leading to us. Now this is what we call an evolutionary tree, because we start with one ancestor, and they kind of branch out into all these different kinds of creatures. Note, all these different kinds of creatures still exist. Okay? We are coexisting with all these different creatures. Lemurs did not turn into humans. They broke off a long time ago from the lineage that eventually led to humans. If we think about this on my color spectrum, it starts to make a little more sense. Well, we have lemurs, New World monkeys, and Old World monkeys all being kind of bluish. So to us, they all look kind of similar. But the great apes, then we start differentiating between them. But really, they're just different points on the color spectrum. That's all they are. And so to say that chimpanzees and humans are drastically different without acknowledging that all of these are drastically different from one another would be a crime. We have to acknowledge that they're all different. So here's a moment of truth. If we take genetic testing to its logical conclusion, your ancestors, 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 blah, 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 ancestor was a primate. These are members of our long-lost family. So if we think about how we fit into this, and how humans even evolved from these primates, we could see a number of different human forms that existed along the way and became extinct, which is why we're the only human type still on the planet. Follow me on this branch for a minute, and just assume that this is kind of like the tree that we just saw. We have the main trunk of the tree, where we have different forms, actually evolving in a pretty linear fashion, but then they start branching off into different forms. Different forms that really didn't live long as far as millions of years. They didn't even live probably 200,000 years for this particular form of hominid. But it started evolving into some fun things that directly leads up to Neanderthals and us. And that only happened, as this estimates, look at that, in the past 200,000 years, which lines up exactly with the mitochondrial and Y chromosome evidence that we just talked about. Another fun thing is thinking about how many of us are part Neanderthal. Because they were coexisting at the same time as Homo sapiens, there was a lot of intermixing between the two. I actually have more Neanderthal DNA at 2.7% than 3/4 of the population. I actually have Neanderthals to thank for not having much back hair. That's awesome. Awesome that they can tell me that. Because I hate back hair. I feel like a gorilla otherwise. But I have these guys to thank. It's really cool. But the main point here is that you did not result from your parents evolving into you. Okay? So to think that we resulted from apes evolving into us is completely false. Okay? Everything is a branched tree that eventually resulted in the population of humans that generated us. So there is no one creature evolving into another creature. It's just a branched tree. Now, one thing you're going to ask is, what about races? Right? All sorts of races that are very, very different in looks and characteristics. Like, well, aren't races evidence of humans evolving into new species? Well, truth of the matter is that humans haven't been around very long. And the other thing to consider is the fact that humans actually interbreed a whole lot. A whole lot. Which is to say, our gene pool has mixed into a worldwide gene pool. And there's actually more differences within any single race than there is between one race and another when we look at the genes. So it doesn't matter if we're looking at Africans or Europeans. There's more differences between me and my wife than there is between me and someone of color. So races are really an artificial construct that we have generated. They're not subspecies. They're not terribly meaningful. And they should never be used as a way to discriminate. Because they're really much more similar than most people know. The whole reason for us having different races really comes down to micro-adaptations to the environment in those different locations. If you're closer to the equator, you're going to have darker skin, because you have much more intense UV radiation there. And what that's going to do is that's going to allow you to produce adequate levels of vitamin D. But if you're further north or further south from the equator, you know, my European ancestry, further north, I don't get nearly as much UV radiation. So I have very fair skin. That way enough light penetrates my skin and I can produce adequate levels of vitamin D. Yes?
So is it like a gene that is switching on and off depending on environmentally where we're at? That a gene would switch on for darker skin, or if you're in the north for lighter skin? Is it environment affecting the genes to switch them on and off?
My problem with that is the fact that that kind of assumes an active adaptation. Everything is passive when it comes to evolution. There's one thing that drives evolution, and that is survival. If you are in a place where there's a whole lot of sun, but you have fair skin, you're not going to survive very well, because your skin is being cooked, and you are not generating the vitamin D that you need to survive and reproduce. So those genes are going to die out. Whereas if you have darker skin, then you're going to have greater levels of survival. And you'll have greater levels of offspring. And that's going to drive evolution in that direction. So inevitably, it comes down to genes turning on and off and regulating high and low, but it's all driven by the need to survive. Everything in evolution is the need to survive. Tied to this is also folate production, which really relates to survival of offspring. So as far as why this is even a controversy, well, it really comes down to people not being very happy that they're told that they come from apes. I sympathize. I hate thinking that came from something that's very hairy. Not a fan of hair. But you know what? That's the way it works. We're told the facts, and whether we agree with them or not, they're the facts. It really doesn't change anything. I hate that gravity acts on me every day. I wish I could just tell gravity to go away so that I can jump 100 feet in the air. That doesn't work like that. Whether you believe in gravity or not, it still exists. That's the same thing with evolution. Whether you believe in it or not, it doesn't change the facts. If we look at humans and how much we vary between each other, it only amounts to about 5% of our DNA. That's a fact. Now, I'm going to talk about dogs here for a second, so bear with me. I'm not the biggest fan of dogs. I know my wife insists on having a dog in the house. But that's okay, because I have snakes in the house, so it kind of balances each other out. But if we look at dog breeds, they vary as much as 28% between different dog breeds. That's a huge variation. So if we think about the differences between all humans, it's at least 5 times as much in dogs. Why is that? It's a very good question. That's because humans, for the past 13,000 years, have domesticated dogs. They have artificially selected for certain breeds of dogs. That has generated this huge variability in their genome, and the huge variability in looking at something that's a Chihuahua versus something that is a Husky. A lot of differences between those two. And yet, more often than not, we have no problem with this statement, that dogs evolved from wolves, that humans are the ones that took wolves and domesticated them. We have no problem with this statement. And yet if we look at it, dogs are only 72% similar, genetically speaking, to wolves. We have no problem saying this. When we have a problem is when we say that we're 95% similar to chimpanzees. That's when we have a problem. Why? I don't know. Additionally, for you dog lovers out there, you do share something with Fido, about 5% of your DNA. So, make you feel a little better about yourself. But no, humans and dogs are really quite a great example because they have co-evolved for the past 13,000 years. And it's really cool to kind of look at some of the different mechanisms that produce them. So with that, I'm going to end and ask you guys if you have any questions, in particular with regards to the primordial soup that I've represented here, just to give you an idea of where your ancestors came from. So, thank you so much.
Yeah, I have a question. Jerry Coyne and others tell us that there never was an Adam and Eve, and that the fewest number of humans that there ever could have been would be like 24,000. Did you know that and how they determined that? Because you didn't say that, but I'm not sure how they determined that. Do you know?
That the fewest number of humans?
Yes, they say science has shown that there never could have been an Adam and Eve because at that point in time when they were supposed to have lived, whatever point they would say it is, there couldn't have been less than, I think the number is 24,000 of those species.
Yeah. More is used euphemistically. The reasoning for that is because of this, right? It's a color spectrum. So we didn't magically one day pop out of a chimpanzee and become human. Right? So that is to say there was a population that was isolated from chimpanzees and went on their own evolutionary path and accumulated enough changes, including the necessary changes to the Y chromosome and to the mitochondria, in order to be considered human. So when evolution occurs, it doesn't occur in just one organism. It tends to occur in the entire population of organisms. And because humans are so widespread, they're so nomadic, that really is why you can't have just one Eve and one Adam. Absolutely. Yes?
Currently, there's a lot of attention being paid to non-binary gender assignment.
Right.
And where does that theory or controversy fit in the whole concept of chromosomes and the evolutionary process? And does it fit in anything you've said here?
No, and I was intentionally avoiding it. That's kind of a talk all by itself, but I'm just backing up to this image just to show you that this is generally how it works, right? Generally, you get one X chromosome from your mom, one X chromosome from your dad in order to become female. But, as I've said before, this is your genotype. That doesn't necessarily make it your phenotype, which is to say the environment, even the internal environment, can shape what gender you actually become. Now granted, all fetuses are female by default. They await a signal from the Y chromosome. But even if that Y chromosome is present, it may not necessarily give the signal. And even if there is no Y chromosome, the body still may get some sort of signal from some source. Now, most notably, that's testosterone, in order to become male, even though this is your genetic profile. So, once again, this is kind of a starter set, the foundation. Whether you actually decide to build on the foundation is another matter entirely. So that's why we have such a gamut, such a wide gamut of what is gender and what is sex. Because even if this is your gender physically, it may not be your gender mentally. And that comes down to hormonal control and its influences on brain development and basically what you desire from life and partners. So it's a really wide, convoluted thing and an interplay between the environment and your genes in order to determine what you are. Which is why when we come to presenting what is male and what is female, in every single way, shape, and form, this is actually closer to being a spectrum of its own. Female over here, male over here, shades of gray in between. Yeah.
Can someone have an extra X? Instead of 2 X's, they have 3, or an extra Y?
And that was something I also intentionally left out because of the fact that most of you are already familiar with a chromosomal aberration, so to speak. And that is with this one. Anybody tell me what happens when you have 3 copies of chromosome 21?
Down syndrome?
Down syndrome. It's trisomy 21. Sometimes the effects are horrible, and the baby doesn't even develop into a child. Okay? The embryo is aborted, more often than not. But sometimes the embryo lives. People with Down syndrome are actually lucky. Most of the time when a 3rd chromosome gets thrown in the mix, the fetus can't develop. It just self-terminates. So these people are lucky. These people tend to be much more lucky and unlucky at the same time, because they can generate very different characteristics. If we have 2 Y chromosomes, then you would expect that person to be a little bit more male in certain areas. But it still just kind of puts the body into this haywire kind of mode where all sorts of weird things happen. It can start affecting your stature, your ability to reproduce. It typically kills that because of the fact that you have too many chromosomes to divide equally and form functional offspring. Same thing here, you can have 3 X's, you can have XXY. That generates a very interesting condition which can also affect how male or female you are. But once again, that's the starting toolkit. That's why when you have certain chromosomal conditions, we can't necessarily predict what's going to happen in you, because it can develop into a couple different pathways based on the environment.
Do these kinds of variations occur in other species, like in the plant kingdom? Can you find plants where this sort of aberration appears, or has not the research been done? I mean, we seem to want to assume a binary condition for everything.
Right.
And is something other than binary prevalent in other species?
Well, the reason why I was pointing to this is because plants are really, really weird, and that's why I really don't like getting into plants, because they can duplicate their chromosomes. Which is to say, if they end up being XXY, they can just duplicate that and then have an even number of chromosomes again, so it divides evenly during meiosis. they could fix their own problems, essentially. That's why plants are weird, and that's why they can get so many chromosomes, because they can do this spontaneously. Most other animals have to deal with it, though, which is to say, more often than not, they become infertile. However, there are cases where this kind of system does not apply at all. Right? In snakes, it's actually the opposite. In snakes, it's the females that have the different chromosomes, and the males that have the same chromosomes. But because of that by itself, snakes have the ability to clone themselves, because they have everything that they need to produce both male and female offspring. So they can spontaneously have babies without being with a male. So they have all the chromosomes that they need. So yes, it does occasionally generate weirdnesses, where you can have an entire population of snakes that are strictly female. And all they've done is clone themselves. Yes?
In your slide where you had identified that we came from a predominant mother and father, so to speak, between 140 and 150,000 years ago. What was the sample size and included in that study? So did it evaluate ancient races like the Mayans or the Egyptians? What were those included in that study?
Typically speaking, it's hard to relate this to one study, because all of this is a conglomeration of information. Every bit of information that we have all originates from these points, which is to say, the Mayans and such, they all irradiated from here. We know that. So we can tie that into all the previous information that we know about where this population came from, which is here. This population came from here, and this population came from here. And that originates from the first humans that we consider to be human. Mayans really just came over, I think the leading thoughts on that is across the ice bridge, across Russia and Alaska. Otherwise, there is some influence from sporadic adventures across the oceans, but really the predominant influence is from the ice bridge. So, to answer your question, yes, we have evaluated other populations, and they all also spawn from this point. So the hub, to be fair, the hub is the Middle East, but it's a hub, it's not the origin. Yes?
Speaking of that, I read recently that they found a new fossil in Morocco that is, if I recall correctly, 7 billion years older than Central Africa. It was in northwestern Africa, still Africa, but it was northwestern Africa, Morocco.
Yeah, and yeah, this is absolutely changing all the time to shape kind of the story that we think of as far as where humans originated from. So yeah, it's still a developing story, absolutely.
One more question.
Yeah.
In determining paternity, you said the strands have to match, they compare mother, the child, and father, and the child. And how many strands matching does it take to determine true paternity? Would you say 13? You said there were only 13 things they looked at?
Right,
Is that the
right. So really what we're looking for is that every band in the child comes from one of two places,
Right.
either the mother or the father. So as we look across here, there is no band that the child has that is not also shared by the mother or the father. That's how we can determine
So
paternity.
it's not how many bands match. It's
No.
If there was even one that didn't match either parent, then we could say that that child's not
Right.
a child of that father.
Yeah. Because generally speaking, we know who the mother is.
So would a grandfather or grandmother have a matching band in that as well?
Generally speaking, we are thinking of something that has such a high variability that the chances of them matching are about 1 in a billion or so. So highly variable sites, they're highly unlikely to be replicated in any way, shape, or form. Yes?
Do we know the location of the origin of life?
No.
Would it be possible to trace that back to?
Unfortunately, the issue with that is that we're really left with fossils. And the problem that we were predominantly a water planet when life evolved in the water, which is to say, very, very difficult to lock down the location of that. So I've not studied that quite as well to see if we've locked that down yet, but the last time I checked, we had not locked down a location on that. Because we're really left with fossils, and tiny things like single-celled bacteria, they really don't imprint very well on a fossil. Yes?
Chris, couldn't that be, given the climate, if you will, at the time of the Earth, it could have evolved, if not evolved, at least, I don't know what the word is, life could have begun in many different places.
Right, so our leading thoughts is that it really needed to require water, just because water is a great medium, it's a great solvent for dissolving other things and bringing molecules together. That's why our bodies are 70% water, is because it's a great medium to bring different things together for chemical reactions and such. So we really kind of need water for life to be possible, which is why when we look at planets that can support life, we typically look for water. Not only for that, but also because of a number of other properties of water that we're not going to discuss. But so, yeah, generally speaking, we're talking about water.
That would be life as we know it. Is it not possible that there could be another kind of life that didn't depend on water, like crystals or something?
So the really important part is a solvent. It doesn't have to be water. It's just that in order for life as we know it to operate, it requires water. That's not to say that silicon-based life could operate or could not operate in a different medium like liquid methane.
Okay.
So the importance is having some sort of liquid medium to bring those chemicals together. Yes?
I understand that viruses are technically not living things. So how does that work?
So I also intentionally avoided talking about viruses because they are really strange little beasties. And the reason why they're so strange is because they are only alive when they're next to something living. Which, if you know me personally, you know that really I'm only alive when I'm next to my wife. Otherwise, I'm pretty much a dead space in the room. So there are definitely cases where we have exceptions to where they act living in certain contexts, and that exception is viruses. Did that answer your question?
How do they transition? What form do they have before they come adjacent to something living and they can reproduce?
So essentially what they're in is in a protein capsule. So pretty much protected from most environmental conditions, which is why they can survive so well on anything, whether it's me sneezing all over this podium or whatever. They typically can survive there very, very well for a long time. And basically what happens is this protein capsule waits until it touches something that's living, and then it starts interacting. Otherwise, it basically lies dormant for a very, very long time.
So it's not a precursor to life.
No. It is its own form of life, given certain contexts. Which is why we're not saying that the first organism had anything to do with viruses. Viruses and prions are aberrations of complex life that evolved much later.
Much later.
Much later.
Okay.
Yes.
Every fall, I'm advised to get a flu shot. And the reason is, and throughout the year,